Hello, I'm Robert, fact checker and science blogger, and this is about the new variant. And first of all, a very simple point. People are getting so terrified about this new variant and saying it's going to spread so quickly and it's going to be such a problem to the world. In the actual data that they're using, which they get this correlation, then notice that the two highest points here, high star node, have close to zero new virus. And indeed it could actually even be equal to zero, because it's a proxy and a certain amount of the old variants also have the same effect in the PCR tests. So whatever is the reason for whether there is a contribution from this new variant or not, for sure, a lot of the reason for the increasing transmission in London is for the very simple, obvious reason that people are not wearing masks, they're not physically distancing. When they get the disease, they don't get tested. If they do get tested, they don't isolate. And if they are in contact with someone who tested positive, they don't quarantine. And if you don't isolate, then you're putting your own household at risk. If you don't quarantine, if you're contact, you put your own household at risk. There's absolutely no doubt at all. There's absolutely nothing to do with the new variant. Those numbers up there, because there isn't any of it. So I just want to make that point very clear right away. And you protect against this new variant in the same way that you protect against all variants of COVID. And you just do the physical distance, hand hygiene, mask wearing, etc., etc. Just do all those things, and you're protected against both of them. Now, this little question that I have, and nobody seems to be looking into it, and so that rather puzzles me. I haven't got any more figures here to help. Yes, here we have. Right, so this is the idea of viral fitness. So the thing is that in these very regions where there's an awful lot of community spread and very high R0, high transmissibility, then you will often get reinfection. So you have people who have been, who got infected, and then it takes 2 to 3 days minimum before you start showing any symptoms. Very few before 2 days. And most people it's 5 days before you have symptoms. Then it's another 7 days from half of them, at least 5 days. Another 7 days after that before you develop the antibodies. So you have a 10-day window for another infection. And these people are just going around, they're just socializing with their friends. Even if they're feeling sick, they're still socializing with their friends. They have lots and lots of opportunities to have a second virus infect them after they've already been infected. And so then this variant would be coming in, and this is an example of something that happened with Ebola, and it's VP30 mute. And this one, if you start off with just over a third with this variant, and then just go through cell lines, and after 9 cell lines, it's nearly all the new variant. So this is inside the human body, faster transmitting from cell to cell. So you've been infected, you go around socializing with your friends, you don't even know you've been infected. You feel a bit sick, you still go on, and then another one of these variants, the second variant, maybe 10 days later, infects you. It infects the upper respiratory tract, where it's going to be, first of all, it encounters the old variant there, but it's got a slight increase. It very much outcompetes it. And then if you go on and infect someone else, then you're going to be infected from the upper respiratory tract. So it could happen very quickly, that this new variant, and it doesn't matter how quickly or slowly, if it's going to, I mean, that's just nine generations of cells of transmission, and you've got nearly all the new variant in the new, and this is with Ebola, and so it didn't need to be more transmissible. And there's another one where that was better at reproducing inside the cell, and this other variant was better at getting into a cell. And neither were necessarily better getting into people, they were just better getting into cells once they're inside people, and they're competing with existing variants inside people. So if that happens with Ebola, why can't it happen with COVID-19? So this is a question I had for the experts there. I haven't found anybody able to answer it. No, I mean, nobody's even tried. Nobody's answered. I've asked several questions on Twitter, and my question has just been ignored. And I can't find anyone on Twitter who is of the researchers discussing this. And I don't know if it's because I'm missing something, and there's something I'm not seeing, and it's something they all know, but then why didn't they answer my question and say, well, we all know this, that's the reason, and nobody's answered it. So I don't know what's going on and why nobody's researching into this, but I just can't see anyone who's researching into this. And it certainly doesn't seem to me at all proven, and the experts are definitely say there may be some element of more transmissibility, but they don't, but it's certainly not all proven that all of this is explained by that. It's not by the high R naught. But I mean, if you had a high R naught, if it's only the way you would be able to test this, if this hypothesis would be that you would, if you search around amongst all the swabs that they've got, and there will be some where the two of them, you see two variants in the same swab, and that would be the moment of time where you swab that person just when new variant is entered and it's not yet overwhelming. And if at that point, if you could go back to the same individual, don't know if that's possible, then you might find that a few days later there's only the new variant. If that happened, that'd be pretty clear proof that they had a second viral infection and that it had overtaken the first one. And so if that's going on in a big way in regions with high community spread, then the mathematical pattern of spread like that would look very similar to the pattern you get from it being more transmissible from human to human. Because that particular variant is more transmissible to other humans, because when it encounters humans with the other variant, it overtakes them. And so if you just follow that particular index case, then it's going to have an R naught much bigger than the other ones, even though the total R naught is the same. So this it's going to look as if this one is the one that's spreading very fast through the population, and then that also would be wherever you have lots of people mixing, and this new variant is going to be spreading more. And so, because it's got many more opportunities, the higher the R naught, the higher transmissibility, the more people are encountering people who are already infected and infecting them again, and that would be the conditions to favour the new variant. So that's just an hypothesis. I have no idea if this is true. I was very much interested to know if any of you know if anyone is looking into this, and if they are looking into this, what they found out, and whether this could be a reason for why this new variant seems to be spreading so much in the population. And if this hypothesis was true, then if you look in regions where there isn't much where it isn't very intense community spread, and where people are, if you look in regions where there's high compliance with masks, there's another way you could look at it without, apart from the extra swab things. You could look at regions where there's high compliance with isolation and quarantine and masking and physical distancing, then you would expect that the new variant to have much less advantage over the old variant, because there's much less opportunity for reinfection. So that might be another way that you would notice it. Anyway, just thought and just upload that. Anybody know anything about that? And just this main point that it's still the same virus, and in the actual data from one of the actual papers that they published on this new variant, and you can see in that data that in the last week in the United Kingdom of their data, then it was the region with just about zero, one of the regions with just about zero of the new virus that had by far the highest reproduction rate, did very, 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 very high. You know, look, that's well above 3. I think that's roughly equivalent to the reproduction. It's called the multiplicative advantage. It was well over 3.5 there, and that had almost none of the new variant. And presumably people being very sloppy there and not doing much to protect themselves or anyone. So, anyway, do protect yourself. You can protect yourself against this. It's not changed. It's the same virus, even if there is some slight difference in the viral load, which is not yet known.